All right, these first couple slides represent our introduction to eukaryotic cells. And the very first thing you need to think of when you think of a eukaryotic cell is what is not a eukaryotic cell. If you'll recall, those are prokaryotic cells. Prokaryote meaning before nucleus, eukaryote meaning true nucleus. Eu means true. And karyotic actually translates to kernel, but the kernel they're talking about is the nucleus of the cell. So if we label the structures in these cells, and you should recognize these cells as onion epithelial cells because you participated in a lab where you were looking at these under the microscope. You made your own slides and looked at them under the microscope. So that little kernel in the middle is the nucleus. Everything surrounding the nucleus, but still within the plasma membrane, is the cytoplasm. You can't see it in this image, or maybe it's barely visible, but there is the largest organelle within most plant cells is going to be a big bag of water with minerals, aqueous solution, known as the central vacuole. Central because it's in the middle of the cell. And notice that it has the effect of pushing other organelles off to the side, like the nucleus. If you look in most of these cells, the nucleus has been shoved off to the side by the central vacuole. Then we have the plasma membrane, and I was trying very carefully to make sure this pointer line reached to the plasma membrane, which is just, again, you really can't see it. It's a thin lining just inside the cell wall, but that's the plasma membrane. And then outside of that is the cell wall. But actually, when you're looking at these lines that are separating these two cells, it's actually two cell walls, right? We've got the cell wall from this cell that's up above, and we've got the cell wall from this cell that's down below. So these lines are actually two cell walls thick. So that's the cell wall. Do you see chloroplasts? These cells aren't green. The reason they're not green is because they don't have chloroplasts, which means they don't have chlorophyll, which is the green pigment within chloroplasts that absorbs the sun's energy for photosynthesis. And it's because this is an onion. Onions are underground. These cells are specialized to be part of the onion, which does not photosynthesize. So this is an example of how plant cells are specialized, just like animal cells are specialized, to carry out particular functions. So the function of this cell is not to photosynthesize. It's to cover. It's epithelium. So the function is to cover, and what it's covering are the layers of the onion. So this is, if you'll recall, a thin, very thin sheet, one cell layer thick between the layers of the onion. So that's why there are no chloroplasts. So where are the chloroplasts in an onion plant? They're above ground, in the leaves. The leaves have cells that are specialized for photosynthesis. They have chloroplasts. Very simply. All right, so the flip side over here, we're looking at an animal cell. And animal cells are arguably more simple than plant cells. Plant cells actually have more organelles, more different structures than animal cells have. So there, of course, is the nucleus, the cytoplasm, everything outside the nucleus but inside the plasma membrane. And the question here is, why no cell walls? Why don't animal cells have cell walls? I mean, you would think that cell walls would be an important thing to have because they really protect the cell and help to support the cell. Well, if you think about the difference between plants and animals, plants are generally stationary. They really can only move from one place to another either through their distribution of seeds or by growing from one place to another. Animals, on the other hand, move. You're not going to be able to move if you're not flexible. So the cells need to be flexible in order to allow animals to move. So I ask you to write captions for these two different images here. So we've got onion epithelial tissue. Plant cells are brick-like, geometric, with cell walls and nuclei displaced by their central vacuole. And again, that's not seen. But just that displaced just means that the nucleus has been pushed out of the way, displaced. These are cheek epithelial cells, just like in the lab, in the cell types lab, you looked at your own cheek epithelial cells. When you did this, you stained them with iodine, but these are stained with another kind of stain that makes them purplish in color, methylene blue stain. They're amorphic, which is a term that simply means they can be just about any shape because they're flexible. There is no cell wall. So notice here that these cells vary in their shape and their size. Because there's no cell walls, they have typically a centralized nucleus. So the nucleus is typically in an animal cell is going to be near the center of the cell. And it is usually the largest organelle in an animal cell. Whereas in the plant cell, the largest organelle is going to be that central vacuole. So animal cells have no central vacuole. They have vacuoles, and we'll learn more about vacuoles later, but they don't have this big, huge vacuole in the center of the cell to store water and other compounds or minerals. So it's the cell nucleus that defines the eukaryotic cell. What prokaryotic cells don't have is a compartment where their DNA resides and is protected because they don't have a nuclear envelope around it. So the nucleus has a nuclear envelope that contains the DNA of the cell in eukaryotic cells. Prokaryotic cells don't have a nuclear envelope to compartmentalize the DNA and separate it from the cytoplasm. That DNA also is associated in eukaryotic cells with a number of different proteins known as histones. So there are a variety of histone proteins that associate with the DNA within the nucleus of eukaryotic cells. And that forms a substance that we refer to as chromatin. And just a reminder, and this is true for both eukaryotes and prokaryotes, there are certain sequences of DNA nucleotides. Remember, that's what spells out the information in DNA. It's the sequence of nucleotides. You can also say it's the sequence of nitrogenous bases or the sequence of bases that spells out the genes. And the genes, whether it be a prokaryote or a eukaryote, will be expressed to make proteins. Remember, that's primarily what DNA codes for, are proteins. But we can also throw in functional RNAs too. Examples being transfer RNA and ribosomal RNA are functional RNAs, and there are genes that code for those. And then we have, of course, the other major compartment within a eukaryotic cell is the cytoplasm. But prokaryotes also have cytoplasm. But it's not separated from what's inside the nucleus because there is no nucleus. So the cytoplasm is everything in a eukaryotic cell. It's everything from the nuclear envelope to the plasma membrane, everything in between the nuclear envelope and the plasma membrane. And it includes the cytoskeleton, which we'll learn more about later, but it's a supportive network of protein scaffolds that run throughout the cytoplasm of the cell, and then cytosol, which is the liquid, the aqueous solution portion of the cytoplasm, and then all the other organelles, and other than the nucleus, are contained within the cytoplasm. And most cell activities take place within the cytoplasm. For example, metabolic processes, breaking down things, building things, take place. Those chemical reactions take place within the cytoplasm. So bottom line here, you and plants have something in common. Animal cells and plant cells have something in common, and that is that they are both eukaryotic cells as opposed to prokaryotic cells without a nucleus. So these represent kingdom Plantae, kingdom Animalia, but also other eukaryotes are kingdom Fungi and kingdom Protista. Basically everything that's not a bacteria, which are prokaryotes. Here we're going to take a look at the structure and function of a plant cell, which is, if you'll recall, a photoautotroph. It gets its energy from light, and it makes its own food. Autotroph means self-feeding. through the process of photosynthesis. And again, it's more complex than the animal cell. So that's why we're looking at the plant cell first, is because there are actually more organelles to be identified in the plant cell than we're going to see in the animal cell. And something to note here is that what we're looking at is a representation of a very generalized plant cell. Not all plant cells have chloroplasts, for example, like those onion epithelial cells. And it's also a caricature. It's important to understand that this is kind of like a cartoon of a cell, where the organelle, the size of the organelles is greatly magnified, is greatly exaggerated, and the number of organelles is greatly reduced. So in a real cell, the organelles are going to be much smaller than what is shown here, and they're going to be much more numerous. They're going to be many, many more. So, for example, when you were looking at Elodea cells, that aquatic plant Elodea under the microscope, you saw that each cell had a lot of chloroplasts in it. Not just, in this diagram, there's only 3. So keep that in mind, that this is just a representation of a plant cell. So we're going to start with the nucleus, and so I'm bracketing the whole nucleus because we're also going to identify some of the components of the nucleus itself. So this whole big blue thing is the nucleus. Within the nucleus, there's this purple region, and it's basically a region, and some cells will have more than one. Some nuclei and some cells will have more than one, but this is the nucleolus. And the nucleolus is the site of ribosome manufacture. In other words, ribosomal RNA components that make up the large and small subunits of the ribosome are made in the nucleolus. So that's its function. Now, that's not going to come up in text here. So I'm only going to label the structures. You need to listen and write down the functions of each of these structures. So again, the nucleolus is the site of ribosome synthesis. Surrounding that is all this squiggly, spaghetti-looking stuff, and that's chromatin, which is, again, a combination of DNA and proteins that we call histones. So DNA and proteins together is what we refer to as chromatin. And this is how it looks. This is how it would look in a non-dividing cell. It's not in the form of chromosomes, right? In a dividing cell or a cell that's preparing to divide, the chromatin condenses into chromosomes. But in a non-dividing cell like this diagram, we're looking at chromatin. And the function of the chromatin is basically the function of the whole nucleus, and which is the function of DNA, right? And the function of DNA is heredity. In a word, it's heredity. But it's to store genetic information and to allow for the expression of that genetic information. In other words, the genes to be expressed to make proteins. It's also that information is going to be copied through DNA replication and passed on to other cells or passed on from one individual to another. Containing the whole nucleus is the nuclear envelope. And it's referred to as an envelope and not a membrane because there's a double membrane. There are actually 2 phospholipid bilayers making up the nuclear envelope. And it's basically a selective barrier. Just like the plasma membrane is a selective barrier, the nuclear envelope is a selective barrier. It's made up of the same kind of phospholipids that make up the plasma membrane that we learned so much about in our previous unit concerning transport, things moving in and out of the cell across the plasma membrane. When it comes to the nuclear envelope, there are some pores, and the term in biology, the term pore simply means a hole, a passageway, a channel. So these are nuclear pores, and those are already in the diagram that you have, but you're going to need to draw in some of these other things that are on the nuclear envelope, and we'll come back to that in a minute, because they're related to or they're involved in the explanation of what all of this is surrounding the nucleus. And one thing to note about this rough endoplasmic reticulum, RER, that's surrounding the nucleus is that it's continuous with the nuclear envelope. In other words, it's connected to the nuclear envelope. That's why it's surrounding the nucleus, is because it is actually connected to the nuclear envelope. And it's rough endoplasmic reticulum because it has ribosomes on it. And that's the thing that you need to draw onto the nuclear envelope also. The nuclear envelope has bound ribosomes, just like the rough endoplasmic reticulum has bound ribosomes. They didn't show that in the diagram, so I wanted to make sure that you knew that the nuclear envelope had bound ribosomes also. And if you'll and we need to back up to the function of the rough endoplasmic reticulum, but it has to do with the function of the ribosomes. So if you'll recall, because you already know this from a previous unit where we looked at protein translation, ribosomes are where proteins are made. Ribosomes are the site of protein synthesis or protein translation. If you'll recall, that's the process of translation. Taking the message that's in messenger RNA and translating it into the amino acid sequence in a polypeptide or protein. That's what happens at the ribosomes. So that gives you some indication that the ribosomes on the nuclear envelope are making proteins for the nucleus. The ribosomes that are on the rough endoplasmic reticulum are making proteins that are going to go into the rough endoplasmic reticulum and be further processed in some way and then transported. So that's the function of the rough endoplasmic reticulum, is to manufacture proteins, modify those proteins, and then ship them out into the cell for either further processing or for use somewhere in the cell. Just like there are bound ribosomes, there are free ribosomes, and I tried to find a good one to point to here, and I wanted it to be close to where we labeled the bound ribosomes. So there are a number of free ribosomes that are floating around in the cytoplasm here. If you look closely, these little dots that are kind of hard to see, because ribosomes are really small, but they're the ones that are just free in the cytoplasm. So those are free ribosomes. And, of course, their function is the same as the bound ribosomes, but the proteins that are going to be made in those free ribosomes are not going to be processed within the rough endoplasmic reticulum or any other organelle. They're going to be good to go in the cytoplasm. So we have rough endoplasmic reticulum, which kind of implies that there might be another kind of endoplasmic reticulum, and that would be the smooth endoplasmic reticulum, or the SER. And it's kind of shown here as what looks almost like a roller coaster that's going back behind the nucleus here. And the smooth endoplasmic reticulum does something similar to the rough endoplasmic reticulum, but it's not for proteins, it's for lipids. The smooth endoplasmic reticulum is involved in lipid metabolism, in other words, making lipids. And really, that's the rough endoplasmic reticulum and the smooth endoplasmic reticulum together is what manufactures most of what makes up cells, because most of what makes up cells is either membrane, that's the SER, or protein, that's the RER. So proteins or membranes made by the endoplasmic reticulum a lot of times are going to be transported to this blue thing that looks like a stack of hollow pancakes known as the Golgi. And it goes by Golgi apparatus, so the whole thing can be referred to as a Golgi apparatus. Each individual pancake here can be referred to as a Golgi body, right? So it's also referred to as Golgi bodies, plural. So it's the Golgi apparatus made up of Golgi bodies. And the Golgi receives packages, vesicles, transport vesicles that come from the ER, that come from either the rough endoplasmic reticulum or the smooth endoplasmic reticulum. Those transport vesicles are going to fuse with the Golgi, and then they're transferred through the Golgi, basically, and as they're transferred through the Golgi, they are modified in various ways. And then they form additional transport vesicles, Golgi transport vesicles, that come off of this end of the Golgi. So there's a transport vesicle right there. And that's going to contain proteins and or lipids that are going to be transported somewhere in the cell for use in the cell, or to the plasma membrane to be secreted from the cell. So this could be a secretory vesicle, as we would call it, because it's destined to come to the plasma membrane and actually be secreted outside the cell. One special kind of vesicle that's made by the Golgi is one that you need to draw in, and that's this. This is representing a lysosome, which is a special kind of Golgi vesicle, a vesicle created from the Golgi, that has digestive enzymes and acids inside it that allow for intracellular digestion of things inside the cell, because there are components of the cell that need to be broken down. And sometimes cells, and plant cells don't typically do this, but sometimes cells take in food. They engulf food from their surrounding environment, like an amoeba. The protozoan known as an amoeba is going to engulf food from its environment. That's how it eats. And that food needs to be broken down. So lysosomes are basically the digestive organelle within cells. Here's the plasma membrane. So there's a cutaway here through the cell wall to show us the plasma membrane. And as you know, the plasma membrane is a selective barrier. So it forms a barrier between what's inside the cell and what's outside the cell, and it regulates what goes across, either into the cell or outside the cell. It selects what is able to go in and what comes out, as you know, because we spent quite a bit of time on a previous unit looking at the function of the plasma membrane and all the transport proteins that may be embedded in the plasma membrane and carrying out that function. If you'll recall, it's a phospholipid bilayer, but the whole model that we use to explain how the plasma membrane, the structure of the plasma membrane, is the fluid mosaic model, where the fluid is the phospholipid bilayer and the mosaic are all the different kinds of proteins that are embedded in the plasma membrane. Then, of course, we've got the cytoplasm, and just like the free ribosomes, I had a hard time finding a good place for the pointer line for the cytoplasm, because the cytoplasm refers to everything here that is outside the nuclear envelope, but inside the plasma membrane. So the whole region that contains all the other organelles besides the nucleus is the cytoplasm. And if you'll recall, the cytoplasm is made up of cytosol, which is the aqueous solution part of the cytoplasm, and all the organelles that are outside the nucleus but inside the plasma membrane are contained within the cytoplasm. And then there is something else that you need to draw in, because it's not part of this diagram, and that represents the cytoskeleton. And you'll notice that there are both thick lines here and thinner lines. And the other thing you need to note about the cytoskeleton, it's not just this little portion in this one place within the cell. It's a network throughout the entire inside of the cell, and it supports, just like your skeleton supports your body, it supports the cell. But it also anchors things, anchors organelles in place, for example, and allows for the transport of things. So, for example, transport vesicles, like the vesicles that come from the endoplasmic reticulum and the vesicles that come from the Golgi, they can be transported along microtubules, which are the large structures here. The fatter lines, the thicker lines, are representing microtubules. They can act kind of like railroad tracks, where special proteins called motor proteins will latch onto a transport vesicle and then latch onto the microtubule and walk along the microtubule carrying the transport vesicle. So things can move from one place to another within the cell along the microtubules, kind of like a railroad system. And then there are microfilaments. These thinner lines are microfilaments. And they are contractile. They can actually contract like muscles can contract, because they're made out of a protein called actin, which is also the protein that's in one of the proteins in your muscle cells that are involved in contraction. So that's the cytoskeleton. Then we have another organelle that you need to draw in, and that would be the centrosome. And it's called the centrosome, centro because it's near the nucleus. Typically it's found near the nucleus, which is near the center of the cell, sort of. Some means body, so anything that ends in some just means body. So this is a central body. It's near the center. And its function is to organize microtubules. So that's why I brought it up here after explaining what microtubules are, making up the cytoskeleton. So the centrosome is involved in organizing the microtubules that make up the cytoskeleton but also, very importantly, organizing the microtubules that are involved in cell division. Because when a cell divides, the chromosomes have to be divided up between the two new daughter cells. And this whole structure called the spindle forms. And that spindle is made out of microtubules, and those microtubules grab onto the chromosomes and separate them during cell division, and the centrosome is involved in organizing all of that during cell division. So it's a very important function. Then we have the mitochondria, but I'm pointing just to this single mitochondrion, so it's a mitochondrion, which is singular. There's another mitochondrion over here, so mitochondria would be a plural. There's another shadowy one here, and these are cut open so you can see the folds in the inner membrane inside. So whenever you see in a diagram kind of a sausage-shaped or a bacterial-shaped, bacilli-shaped, like a bacterium, or elongated structure that has a squiggly line going through it, that's a mitochondria. That's how you can identify it. And mitochondria, in 3 words basically, the function of mitochondria is aerobic cellular respiration. That's where the metabolic process of aerobic cellular respiration takes place, which briefly is the breakdown of organic compounds to make ATP. So, for example, to break down glucose, to make adenosine triphosphate, ATP, the energy currency of all living cells. Then, of course, this is a plant cell, so we have to identify the chloroplasts. But again, not all plant cells have chloroplasts, only the ones that are specialized for photosynthesis, which is one of many plastids. So, turns out chloroplasts, notice that plastids, chloroplasts are just one kind of plastid that you'll find in plant cells that are carrying out a variety of functions. Other plastids, for example, either manufacture specific things or store specific things like starch. And we're not going to get into the details of what different plastids there are, but I just want to make sure that you understand that chloroplasts are just one of a whole family of organelles within plant cells that are known as plastids. And just a reminder, too, that both mitochondria and chloroplasts within eukaryotic cells are thought to have originated as bacteria that became what they are today, mitochondria and chloroplasts, through the process or the theory of endosymbiosis. So I just wanted to remind you of that. The origin of eukaryotic cells in general is thought to be through the process of endosymbiosis, which is a concept that we've already covered, but we're going to cover it again in this unit because it is so central to the evolution of eukaryotic cells. Finally, again, because this is a plant cell, we've got this big sausage-shaped bag of water basically in the middle of the plant cell. That's the central vacuole. Central because it's near the middle of the cell, and it's a giant vacuole, which kind of sounds like vesicle. So, and here you can kind of see the difference between vesicles and vacuoles. One of the differences is that vesicles are much smaller and they're specialized for transport, whereas vacuoles are larger and specialized for storage. And that's the function of the central vacuole. It primarily stores water with minerals and other compounds dissolved in it that the plant wants to hold on to. So generally its function is simply storage. But it has a secondary function because the water that's in there is under pressure. Because of the osmosis of water into the central vacuole, it increases the pressure within the central vacuole. And we call that turgor pressure. So it's a special kind of osmotic pressure, turgor pressure, that helps to support the cell. That pressure is why plants are able to stand up. And if you don't water the plant, then the pressure within the central vacuoles decreases, goes down, and the plant wilts. So its primary function is storage, but its secondary function is to support the whole plant and support the plant cell. Finally, we have the cell wall. And the cell wall is for structural support. It's not to control what goes in and out of the cell. You can think of it as kind of like a screen door where the air can go right through it. pretty much anything can go right through it. However, there is some protective function. For example, viruses have a hard time infecting plant cells because of the cell wall. So, and bacteria too are going to have a hard time, because bacteria are even bigger than viruses. They're going to have a hard time getting to the plasma membrane and infecting the cell. So its primary function is protection, but also structural support, and that's why plant cells often have this geometric shape. And in this diagram, they're showing the cell wall from an adjacent cell. In other words, a cell that's next to the cell we're looking at here. It's only showing the cell wall here, just to let you know that cell walls come up against each other between two plant cells. But there are these channels, holes, that connect the cytoplasm of two adjacent plant cells. So because the cells themselves can't move within the body of the plant, the cells tend to be connected to each other with these special channels that we call plasmodesmata, which is nothing I'm going to hold you responsible for. I'm not going to label it. I just wanted you to know that there is a name for those plasmodesmata channels between plant cells that connect the cytoplasm between the cells. So finally, what's the difference between plant cells and animal cells? Well, again, plant cells are more complex. They have more structures than animal cells, and that's why we're looking at them first. They have all the components that animal cells have, plus cell walls, a central vacuole, chloroplasts, and other plastids. So that's it for identifying the structure and function of a eukaryotic cell. Now, on the slides coming up, we'll be looking at each of these different organelles individually to take a closer look at their ultrastructure, the molecular makeup of those organelles, and again revisit their functions. But before we do, I just want to give you an overall view of the evolution of plants. So remember, plants and the chloroplasts within plants are related to cyanobacteria. These chloroplasts are very cyanobacteria-like. And so through the process of endosymbiosis, plant cells now have chloroplasts. Serial endosymbiosis, because if you recall, there were two endosymbiotic events. First, taking on mitochondria, and second, taking on chloroplasts. So that's the theory that tries to explain where mitochondria and chloroplasts came from in eukaryotic cells, and tries to explain the evolution of eukaryotic cells in general. So then we have what would be algae, the first eukaryotic cells to contain chloroplasts would be considered algae, and they would be unicellular algae. Some unicellular algae then evolved to become multicellular algae, and we'll see that when we study algae in this unit. And then some of those multicellular algae, which live in water, gain the ability to live on land, and that's what we call a plant. All right, so that's basically, in a nutshell, the evolutionary sequence that arrives at those eukaryotic cells that we call plants today.